Assalamualaikum and a very good day to my lecturer and all my classmates. Today we're going to present about transpositions and illegitimate recombination. Our members is me, Nur Farahanani binti Muhammad Yusuf. Second is Nurul Shazwani binti Muhammad Kamarul Arifin. The third one is Nur Fatin Nazihah binti Roslan. And the fourth one is Nurul Anis Aribah binti Zulkifli. The contents of our presentation is transpositions and illegitimate recombination. The important point under transposition is transposable elements, mechanisms of transposition, disease caused by transposition. Under illegitimate recombination, the important point is mechanisms of illegitimate recombination, disease caused by slippage recombination. First one is transposition. Before we move on to transpositions, we need to know what is transposable elements is. It is a specific sequence of DNA that codes for enzymes for their transpositions from one position to another position in the chromosome. It is also known as transposons or jumping genes. Discovered by Barbara McClintock through an analysis of genetic instability in maize. She earned her Nobel Prize in 1983. Broadly classified as insertion sequence (IS) or transposon. Insertion sequence are the simplest types of transposable element with short sequence of about 1000 base pairs present in bacteria and code on the genes for mobilizations and insertion. There is a diagram that I provide to visualize the insertion sequence. Next is transposons, which have the longest sequence of more than several thousand base pairs. It contains additional genes such as antibiotic resistance. I provide the diagram below for you to visualize what is transposon is. Transpositions is a process in which a transposable element is removed from one site and inserted into a second site in the DNA. Transpositions of genes is linked to the presence of transposable elements. There are two mechanisms of transpositions. The first one is replicative and the second one is non-replicative. For the first mechanism, which is replicative transpositions is a direct interaction between the donor transposon and the target site, resulting in copying of the donor element. It is also known as copy and paste transposition. Transposition is catalyzed by an enzyme transposase, which is encoded by the transposable element itself. It presents only in prokaryotic organisms. This diagram shows the process of replicative transpositions. In this type of transpositions, a new copy of transposon, which you can see in the purple one, is introduced at the new site. In order for transposon to be inserted into the new site, the enzyme transposase will cut the target site highlighted in red. After that, the transposon will be inserted and the gap will be filled by the DNA polymerase. The second mechanism is non-replicative transpositions, involve excisions of elements and reintegrations at a new site. It is also known as cut and paste transpositions. This type of transpositions is catalyzed by an enzyme called transposase, which is coded by such transposable element itself. Conservative transpositions is present in both prokaryotes and eukaryotic organisms. Conservative is another name for non-replicative transpositions. This diagram shows the process of non-replicative transpositions. The first step is transposase recognizes and binds to the transposon's 3' ends and 5' ends. Then transposase excites the transposons, which you can see in the green one, from its DNA location. Next is transposase then move the transposon to another DNA locations or to the target DNA. The target DNA you can see in the blue color. Next is transposase clip the target site, leaving sticky ends on both sides. Transposase inserts the transposons into the new locations, and DNA polymerase 1 and DNA ligase repair the single-stranded flanking, result in the direct repeat formations on either sides of inserted transposons. That's all from my part. I will hand over the presentations to next presenter, which is Ariba. Thank you.
So I'm going to continue on the differences between transposition and site-specific recombination, as we already learned about site-specific recombination in the previous group's presentation. So first, transposition involves DNA replication, whereas site-specific recombination involves precise breakage and rejoining in limited regions of homology shared between specialized recombination sites and does not involve replication. Although elements that insert into a target DNA via transposition and via site-specific recombinations are both direct repeats of target sequences, the origins of these repeats are quite different. In transposition, target sequence duplications arise from DNA replication. By contrast, in site-specific recombination, the substrates together contain two copies of the sequences that will be appearing the product as target duplications. Another feature of transposition is that it's non-reciprocal due to the obligatory involvement of replication, whereas site-specific recombination, a reciprocal form of recombination, occurs by breakage and joining without nucleotide loss or gain. So, how does transposition cause diseases? Bear in mind that transposons are mutagens, thus they can damage the genome of their host in various ways. First, a transposon or a retroposon that inserts itself into a functional gene will most likely disable that gene. Then, after a transposon leaves a gene, the resulting gap will probably not be repaired correctly. Hence, multiple copies of the same sequence can hinder precise chromosomal pairing during mitosis and meiosis, resulting in unequal crossovers, which is one of the main reasons for chromosome duplication. So currently, the transposons in our genome are set to their harmless positions and have generally lost the capacity to transpose. However, exceptionally, some transposons can move from the beginning when forming reproductive cells and become integrated inside some genes, altering their expression. This can give rise to certain diseases. In this case, is such as some type of hemophilia, hemophilia A and B. And what's more, they may occur in some cancers too, which are varieties of colon or breast cancer as a result of transposons integrating in key genes of adult somatic cell. Other diseases caused by transposons are porphyria, Duchenne muscular dystrophy, and severe combined immunodeficiency. Transposons have been an especially useful tool in plant molecular biology. Researchers use transposons as a means of mutagenesis. They use transposons to identify the mutant allele, to study chemical mutagenesis methods, and also to study gene expression. I guess that's all for transposition part. I'll pass the illegitimate recombination presentation to Wani.
Thank you, Anis Ariba. Now, Fatin and I will continue on the illegitimate recombination. Firstly, illegitimate recombination, also known as the non-homologous recombination, is actually referred to a pathway that will repair a chromosomal double-strand break in the DNA, especially in somatic cells. The illegitimate recombination will repair this DNA break by joining two unrelated double-stranded segments of the DNA, and some of which will cause the formation of novel DNA joins. It also related to the formation of spontaneous deletion. Franklin 1971 has proposed two different mechanisms for illegitimate recombination, which are cutting and rejoining, as well as the slip Miss Perry. Cutting and joining. Double-strand DNA break are actually common event in eukaryotic cells, and it was estimated there are 10 DNA break per day in each cell. So there are two major pathways of DNA repair, which are homologous recombination, which have been presented last Friday, and non-homologous DNA enjoining and HEG that will be presented today. That's I will be present. And HEG not only crucial in repairing pathology double-stranded DNA in chromosomal translocation, however, it's also important during the variety, diversity, and joining recombination, as well as the class switch recombination. This is the reason when people that lack with normal and HEG will sensitive to ionizing radiation and also suffer in severely immunodeficiency. And this NHEG also an example of natural deletion mutation. Non-homologous enjoining is a mechanism to repair double-strand break in DNA without the need of homologous template. Three important steps in this mechanism, which are recognition and processing and also ligation. The important enzymes involved in this mechanism are Q70 and Q80 in the recognition process, DNA PKCS, Artemis enzyme in the unprocessing process, DNA polymerase mu, DNA polymerase lambda, DNA ligase 4 along with the XRCC4 in the ligation process. Now I will explain in detail the non-homologous enjoining repair mechanism. First, we will have the double-stranded DNA that break. This is the non-homologous DNA. And here we have the non-homologous enjoining repair and also homologous recombination repair. So we begin. The first step is the recognition. The first enzyme will come is Q70 and Q80, which will bind to the both end strand of the double strand break. And then the DNA PKCS will come, then form complex with the Q70 and Q80 here. This complex will cut the nucleotides and produce the blind end. Then it will proceed in the unprocessing step where this complex will start to move in opposite direction by the help of Artemis enzyme. They will start to produce, they start to move opposite because they want to produce the overhang. The overhang will produce is like this. Yeah. Okay. So it's the overhang that will be produced. And then the DNA polymerase mu and also DNA polymerase lambda will start to add on more nucleotide on this overhang to produce longer strand. And then this let then this will be this strand then will be joined together by using the SRCC4 and also ligation 4. Then I have a short video here. The first come is the Q70 and Q80, and then the DNA PKCS will come. They will start to cut, and then they will produce the blunt ends of strand. Then the DNA polymerase will come and produce more, add on more nucleotide to produce more strand, and then the XRCC4 and ligation 4 will join the overhang together. The difference between NHEG and also HR are in non-homologous enjoining, the repair is intrinsically mutagenic, which will cause the deletion mutation. However, homologous recombination is produce the accurate repair. and HEG will process in very fast mechanism like estimate 30 minutes. However, in homologous recombination, there are much slower, like 7 hours and longer. And for the homologous enjoining, they are active in all phases of cell cycle, while in homologous recombination only active in S and also G2 phases. That's all from me. Thank you.
Hi, my name is Nur Fatina Jamiti Roslan with metric number 198778. So now I will talk about the second mechanism of illegitimate recombinant, which is slipped mispairing. So slip mispairing in DNA occur during DNA replication. When DNA polymerase slipped off from the template strand, it caused the replication to pause. So this will lead to the dissociation of the newly synthesized strand from the template strand. So in the picture we can see the newly synthesized strand is the one with orange color and the template strand is the gray color. Notice that in the strand the sequence are the same, GATA, GATA. So the special thing about slip mispairing is that in the DNA strand the sequence is repeated more than once. So it produces a repeated sequence, and this repeated sequence can occur 2 times, 100 times or more. It varies, depends on each individual. So when the dissociation of the newly synthesized strand of the template strand happen, it will lead to mismatch pairing of the sequence. Since they are repeated and they are complementary with each other, they easily bind at any position, forming bubbles on the strand. So when replication continues, it notices the mismatch that occur, and it will subject to the mismatch repair system through contraction or expansion. So next, so during DNA replication, as I said, the DNA polymerase will slip off the DNA strand, and it fails to copy the sequence that will lead to deletion, or it repeatedly copies a given sequence, producing more repeated sequence. So there are 4 classes of repeats. First one is close repeats, which is a short repeats of 8 to 10 nucleotides that are separated by spacer of nucleotides. Second is spacer interspersed direct repeats. It is a multiple copy of close repeats being separated by non-repeated nucleotides. Third one is simple sequence repeats, which is the tandem repeats of small motifs 1 to 5 nucleotides. The last one is tandem repeats. It is much larger. It's a tandem repeat more than 5 nucleotides. Okay, next we will look at how slippage DNA occur and how they repair this mismatch. Okay, during DNA replication, here on the top we have the template strand, and then replication occur as usual. When DNA polymerase is replicating this template strand, it encounters a region of repeated sequence. This repeated sequence is unstable, causing the DNA polymerase to slip off from it and then lead to the dissociation of the newly synthesized strand from the template strand. Okay, next, it will bind to the repeated sequence, to close repeated sequence at any position, causing it to form bubble. So from here, it will produce 2 outcomes. The first is when the bubble is formed at the template strand. Okay, on the left side, the bubble is formed at the template strand. Okay, when DNA replication continues, it notices this mismatch, and it will repair and restore this strand using deletion or contraction. So it will delete, in this picture, it will delete two frame shape product and causes the complete DNA strand to being contracted from the original strand. On the right side, where the bubble is formed at the nascent strand, at the newly synthesized strand, and this will subject to mismatch repair system, using expansion. So what happened? It will add 2 more frame shift and causes the newly strand of DNA to be slightly longer than the original. So next, slippage DNA can cause many diseases, and here are some of the 2 diseases. So first one is Machado-Joseph disease. It is an autosomal neurodegenerative disease that can be inherited. So individual with this disease lack of muscle control and coordination of upper and lower extremities, includes the arms, hands, legs, and foot. So this disease happen because of the expansion of abnormal CAG trinucleotide that be repeated in the ATXN3 gene that are encoded for ataxin 3 protein. Ataxin 3 protein involved in a mechanism called ubiquitin proteasome system that help to destroy, eliminate, damage or excess proteins. The second is Huntington disease. It is a progressive brain disorder. So people with this disease unable to control their movements, they have emotional problems and also loss of thinking ability. So these people find it hard to engage with conversation and some of daily activities. So this disease caused by repetition of CAG trinucleotide from 36 to more than 120 times repeated in the strand at the HTT gene. HTT gene is encoded for Huntingtin protein. Okay, Huntingtin protein involved in chemical signal transportation of materials and protecting cells from apoptosis, and they can be found mainly in brain. Okay, that is all from my part. Okay, so the conclusion for transposition, there are 2 types of transposition, which are replicative and non-replicative transposition. And transposons can cause a few diseases such as cancer, porphyria, and hemophilia. And there are various applications transposons can be used. Next, for illegitimate recombination, there are 2 methods in illegitimate recombination, which is cutting and joining, and also slipped mispairing. And diseases that can cause by slippage DNA are Machado-Joseph disease and Huntington disease. That is all from us. Thank you.